Bismillah ar-Rahman ar-Rahim. I would like to welcome you all to our 13th lecture in Microbiology Made Easy series. In today's lecture, I will be covering part 1 of bacterial genetic lectures. In part 1 of bacterial genetic lecture, I will be talking about the bacterial genome. At the end of this lecture, I will be grateful to you if you could share this lecture with your friend and colleagues in order to get the reward from Allah subhanahu wa ta'ala. So what is the bacterial genome? The genome of any organism, it could be bacteria or it could be a human cell, defined as the totality of its genetic material. So all of the genetic material of an organism, we call it the genome. Okay, let's look at the bacterial genome. As you see here in this diagram, this is a bacterial cell. Okay, so the bacterial genome consists of the bacterial chromosome, which I'm pointing at with my laser pointer. This chromosome, which is made from double-stranded DNA, it carries all the essential genes and non-essential genes, and also part of this bacterial genome is the bacterial plasmid, which I'm pointing at with my laser pointer. So bacterial genome consists of a chromosome made of double-stranded DNA. Okay, this chromosome will be carrying essential and non-essential genes, and in addition to that, in addition to the chromosome, bacterial plasmid, which is a circular extra chromosomal DNA, which I'm pointing at with my laser pointer. So both bacterial plasmid and the chromosome, we call it total bacterial genome or bacterial genome. As I mentioned in the previous slide, that the bacterial chromosome, it contains double-stranded DNA. Double-stranded DNA arranged in what? In a circular form. So we have a double-stranded DNA arranged in a single form. So you see it here. This is the bacterial chromosome, and it's coiled or occur in a circular form in order for it to be packaged in the bacterial cell. If we magnify the bacterial chromosome, it will consist of, as I mentioned previously, double-stranded DNA. Double-stranded DNA, as I'm drawing now. Oh, you see it here? So this is the double-stranded DNA. So when we magnify this piece of the bacterial chromosome, you see the double-stranded DNA. So this is one strand, and this is the second strand of the bacterial genome. The length of the bacterial chromosome, its length, you can see here, the length of the bacterial chromosome, it's about 1000 microns. Also, bacterial DNA or chromosome, it contains from 2000 to 4000 genes. And as I mentioned in the previous slide, some of these genes are useful for bacteria. Some of them, they are non-useful for the bacterial cell. This, my friend, will bring me to the end of part 1 of bacterial genetic lecture. In the next lecture or part 2 of bacterial genetic lecture, I will be covering the pathogenicity island as a crucial player in bacterial genetic. At the end, I would like to thank you and to ask you to share this lecture with your friend and with your colleagues in order to get the reward from Allah subhanahu wa ta'ala. Thank you.